Hello and welcome to this week's broadcast of This Week in Science. We are back again and hope you're here with us. Oh, people are counting down in the Discord. We're a little bit ahead of you, but not by much. You're going to be catching up in no time. Are you ready for a good show?
Yeah, let's do it.
Okay, let's have a wonderful show. Why does it do this every time? This is...
What happened?
This
We didn't get 2 seconds into the really good show. We ran into some problem. I don't even know what it is.
It's like every week now, even though I'm starting within the Facebook window of time, it says, no, we're not going to let you do that. And so now I'm going to do this again. Hopefully it'll go, Facebook is live. Dun, 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 dun. Fingers crossed. But anyway, we're here. You're there. Let's science it up. If you are watching live, you're watching this stuff. If you're going to subscribe to the podcast, this will not be included because this will all get edited out and other beeps and bloops for the podcast version. But make sure you click the likes, the shares, the subscribes, wherever you are watching or listening right now. And we are so glad that you are here.
Yeah.
Ready to go. All right, let's make this happen. In 3, 2, this is TWIS. This Week in Science, episode number 942, recorded on Wednesday, September 6th, 2023. How much science did you inhale? Hey everyone, I'm Dr. Kiki, and tonight on the show we will fill your heads with a rant reversal, a brainy dish, and less pollution. But first...
Disclaimer. If you have tuned in to this show, you know what you're in for. This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Justin. Welcome back to another episode of This Week in Science. We're so glad everyone is here to join us for another fun-filled discussion of the weekly science news. Yeah, you ready for this, Justin?
I am.
Ready. We're really ready to do this week. World's shortest disclaimer. And let's go.
Yeah, well, sometimes I go on and on and on to disclaimering, talking about stuff. This time I figured, you know what? This is like, I don't know, were we at the 1,000th episode yet?
Not
We're
yet.
getting in there. We're getting close.
We're pretty close there. Yeah, but there's always more science to talk about.
People have heard all of the disclaimers. How many times can I just explain that we're going to do a science show?
It's always different, which is always great. What do we have for the show this week? I have news stories about some embryos in a dish, brain in a dish, some a genetic protection factor, potentially some less pollution, and baddie boxes, good or bad. We'll see. What did you bring?
I've got magic mushrooms for major depressive disorder, how chronic fatigue might be a post-viral infection, and Homo erectus balls. Plus, an update to the 2 week ago speculative rant prediction about Denisovans. I've already thrown it out, and I've got a replacement for today.
Well, that's what we're here for. We get to talk about all of these thoughts in our brains. And if you enjoy all these thoughts in the brains and the discussions that are going on, please make sure that you subscribe to This Week in Science wherever you're watching, YouTube, Facebook, and Twitch. Wednesdays, 8 p.m. Pacific time ish is about when we start. We go for a tight 90 minutes for sure. Ish. We are podcasting just about every place podcasts are found. And if you're looking for more information, you can find our website, twis.org. It's where our show notes and other subscribe buttons and things are. Now it's time for the science. Start with some embryos.
Yeah.
We'll start at the very beginning of, I don't know, human embryonic development.
Exactly where I started.
Researchers at the Weizmann Institute of Science have been looking at how we can take human stem cells, so like adult skin cells that have been reversed to a younger state, pluripotent stem cells, embryonic stem cells, how we could put them all together into a little clump of cells that develops just like a regular human embryo. So no egg, no sperm, no fertilization, just a clump of cells in a dish that organizes itself and grows just like an embryo. And they just reported in Nature this last week that they were able to get a complete model of human embryos from stem cells cultured in the lab and got them to grow to day 14. And we've talked about this.
Yeah, day 14, it's not just that they couldn't go to day 15 because they hadn't gotten the cells right or something. That's the arbitrary cutoff date for how long you're allowed under law, I guess.
It's kind of an international science agreement.
Yeah.
It's a pact, a handshake, a signature on a line. Yeah.
You're only allowed to take them to that many days. Past a certain point, too. There's also like, it's actually right where it's an ish because there's days, but then there's also like, well, if these lines appear in the cell formation, then we also you have to
abort because then it has gotten to a certain state of development that you're not going to go past.
So you can't cheat and have an accelerated development, right? that in 14 days gets beyond this point. There's a cutoff.
Right. We're not speeding it up within that. It is what is supposed to be the natural progression of development during that first period of time. The researcher, Dr. Jacob Hanna at the Weizmann Institute of Science says there's really like exciting stuff happens in the first month. So we're still there. The agreement is still stopping it well before the first month, but like the first month is when like the drama of cellular division takes place. A lot of the problems that lead to miscarriages and other birth defects, those occur or they start to go, cells start to apoptose or be developed in the right way or the wrong way to lead to a viable embryo that can lead to a viable pregnancy. But we don't really know what's going on there. And the ethical way that they want to do this is without embryonic derived stem cells, but adult induced pluripotent stem cells. So the majority of these cells that were included in this were induced pluripotent stem cells. So they reprogrammed the cells. So they went back to this very, very early state called the naive state where they can be anything. They're naive. They can do anything. And they've been working on this since about 2013, and we've reported on some of the stuff coming out of this lab and others related to this stuff. They took the cells and they divided cells into three groups. They had cells that were treated with chemicals so that there was no genetic modification to turn on certain genes. So they were trying to basically induce these naive cells to become different types of tissue so that some of the cells became embryos because they got certain chemicals. Some of them became placenta. Some of them became like yolk sac. They needed stuff to sustain the embryo so that potentially if someday that 14-day limit is taken away, or we know that there are lots of birth defects and other issues of pregnancy related to the development of the placenta itself, if those are understood, then we can really figure it out. The researchers say an embryo is self-driven by definition. We don't need to tell it what to do. We only must unleash its internally encoded potential. And what they've created now is this stem cell-based embryo-like structure. It developed normally outside a womb for 8 days and reached a developmental stage equivalent to 14 days in human embryonic development. So they're very excited because they got placenta, they got yolk sac, they got bits and pieces of all the cells that they haven't really been able to get before. This is the most complete structure that's been viable all the way through the time period that they have that they've been working through. And so this could lead to new directions in pregnancy research, into understanding how the little tiny ball of cells that becomes a human actually gets its start, and how the yolk sac is involved. What are the genes that are involved in making it all work? And so it's very exciting to these researchers and now to the general community working on human embryonic development.
Yeah, this is why I need my own country.
Why do you need your own country?
So that I can not participate in the 14-day rule. So that I can just be like, okay, I've got this small island nation. We're only going to be here apparently for a couple more years because of global warming and sea level rise. However, in the time that we're still above sea level, we're going to authorize that we are not going to participate in the international agreement. And we're going to allow scientific research to go to 28 days at the very least, right? Like, come on, people.
Well, I think this is great what they're doing so far. Everybody's doing protocols according to international agreement, which is fantastic. If the researchers and countries decide to do it differently moving forward, understanding the research and how induced pluripotent stem cells can be used to understand this stuff, it's great. I mean, what they've already discovered with this study is that they need to have placental forming cells at like day 3, like really, really early. The placental forming cells need to be there and viable as their own type of cell really early. And if not, then the yolk sac fails to develop properly, and the embryo will fail. So this is really helpful early stage embryonic development. It's very important to understand. So this is pretty cool.
Yeah.
Answering basic questions about how do we get our starts? How does it work? How do the cells work? What do they need? It's pretty awesome. What do you want to talk about now? You're depressed because you don't have your own country.
Yeah, I'm so depressed for my own country. Okay. This is actually a pretty big study in that it's 34 different researchers from 18 different institutions that run the gamut of like really prominent schools and institutions across the nation. They have looked into using psilocybin, magic mushrooms, in association with major depressive disorder. So they had, this is sort of we've been hearing about these sort of one-off pre-clinical trial sort of proof of concept studies where they have done something similar, had psychological support along with psilocybin administered to see what effects these would have in a therapeutic setting. They've been positive. They've shown really good results on a small scale. So this is a larger scale study And basically what they've done with 104 people this time, as opposed to just a few at a time, 104 adults diagnosed with major depressive disorder were given a single dose, 25 milligram dose of psilocybin, and of course had the psychological support available as well. And they found that they had almost instantaneous reduction in symptoms.
Wow.
Now, that's at the 8-day, and then again it tested at 43 days. That is basically instantaneous when you are comparing it to most antidepressive drugs, which have a scale of several months. It might be 4 months before they will determine whether or not something is working.
They're really working. Yeah.
All right. So really fast start. The other thing that was interesting too is that there weren't any. This was single dose, so this is not daily pills.
Right.
This is not daily intake, which means the actual drug in the system clears, and the patients are still maintaining this reduction in symptoms.
So what that means is that it has, in a single dose, changed or impacted the plasticity of the brain. It's changed the way that neurons are connecting with each other, because that's one of the big things related to major depressive disorder is the brain's destruction or deletion of connections within the brain. And it's like over time, those doses of SSRIs and others are hopefully enabling those connections to rebuild.
Yeah, so they used a couple of different scales, and it has, everything in depressive symptoms that are from anxiety to depression to, the whole thing. And it was, positive results. So the thing to compare this to is the antidepressants, which, like we were saying, has maybe 2, 3, 4 months before they start to kick in after taking daily doses. Mm-hmm. There's an average relapse rate, meaning they stop working within a year. Within a year after that they kick in are working. Mm-hmm. That actually is only effective in about 2/3 of the patients. There's still a third that each time, I guess, that it doesn't work. And then there's about a 50% chance or more that they'll stop working. That they worked for a while, and now whatever the effect you were getting from the antidepressant no longer works.
Yeah.
So so what then takes place is doctors go through a list of antidepressants. You tried this one for 4 months, no effect. We'll try a different one. That one works great. It worked for 8 months, and now it's not working. Okay, fine. We'll try a different one, and you'll have to wait 4 months to see if that works. So you have this sort of constant treadmill of waiting for a drug to work, then it works, and then you hope it keeps working.
Yeah.
Now, one of there's a lot of side effects that come along with these. One of which is an emotional blunting that's associated with a lot of antidepressants. They did not find that with the psilocybin dosing. They didn't experience that major. The other one is withdrawal symptoms.
Right.
Withdrawal symptoms.
Stop taking one. Yeah.
Yeah. The withdrawal symptoms are pretty intense. Like I had to. Here it goes. Discontinuing antidepressants, even if ineffective, can have severe and debilitating withdrawal symptoms, including dizziness, nausea, paresthesia, headache, feeling abnormal anxiety, suicidal ideation, insomnia, and depression. Is a withdrawal symptom from an antidepressant, even if it didn't work. And now you put that into how many of these withdrawal phases are people going through in the search of one that even works?
Yeah.
Anyway, of course, so this research is sort of confirming past research. This time it was a double-blinded, very, robust experimental design, larger cohort, confirming past findings in smaller studies, contributing to a growing body of evidence suggesting that psilocybin is a potential intervention for major depressive disorder. However, it is unlikely to get approved. And the reason is, this is just me talking.
That's you. Yes, this is just
me
you
talking. This is not from the study.
Yeah, okay.
It's not likely to get approved
Disclaimer.
It's going to take 30 drugs off the market that people take every single day.
And not necessarily, though, because we don't know. I mean, this is a number of subjects, but again, it's not all subjects. Not everything works for everybody. So,
that's true. That's true.
I mean, this is it could potentially be another tool in the basket. And, yes. As in this study, they're doing synthetic psilocybin and not natural psilocybin.
Yeah, but
handing people mushrooms. So there's probably someone who has
the difference is the good part's the molecule, right? So that's the thing but the thing is, this is also a testing one dose.
Right.
So additional dosing, they don't come to the conclusion that this is a curative response. They're not looking at that. They're looking at reductions of symptoms and comparables to antidepressants. So
right, you always have to look at like the end point of the study. Yeah.
Yeah, and it's a 43-day study, basically. So they're not going beyond that. This is just another iterative step that science takes. But really, really encouraging.
It is really encouraging. And I, what I also am going to be interested in seeing is there are a number of researchers who are trying to figure out if they can create compounds like psilocybin or ketamine that seem to have these antidepressant effects, but see if they can create chemical analogs that don't stimulate the hallucinatory impact. So maybe the physiological impact is different. So that maybe because psilocybin itself can have negative side effects from those doses. Maybe there are chemical analogs that can work in the positive way without having to have what some people think are positive side effects, but many others think of as frightening and negative side effects. So,
and it might be that those exciting or frightening experiences are part of why they work. Like it might be just part of the process. They might not be able to be separated. In which case, it is difficult because it's like, I only have to take this pill, once a month, but I can't leave the house for 2 days. Then it's like, well, is that going to fit into a busy modern lifestyle or not?
Well, maybe it's not 2 days at home, and maybe it's just a few hours that your, psychedelic therapist guiding you through whatever the treatment is. And
that sounds like the kind of.
There are people working on this stuff, which I think is very cool.
Sounds like the kind of we'll sign up for something. Anyway, psilocybin. Like, what was the other one that we had a while ago? Like, it seemed to improve. It was a one-off, but it seemed to improve color blindness too, for people who still had the minor version of color blindness.
I don't remember that one at all. But
it sounds like you're saying, it sounds like it's doing a rewiring, and that's why it's maybe having this lasting effect.
But how does it do?
It's kind of hard to come up with a product.
Yeah, how does it do that?
It's kind of going to be hard to come up with get a drug company behind a product that they would sell once, and then the patient is like,
I'm good.
Yeah, fine.
Well, we need a different model for how we do things. But okay, there are other health issues that people are up against in their lives as we age. Alzheimer's and Parkinson's are disorders of the brain and nervous system that uh, impact a large number of people on the planet, hundreds of thousands of people, millions of people. But researchers out of Stanford University have published a paper in the Proceedings of the National Academy of Science on their work on a particular gene variant that is called DR4. And it's an allele, so it's like a one version of a particular gene that seems in earlier studies they found it to be protective for Parkinson's disease, against Parkinson's disease. And in this study, they did a big database search related to Alzheimer's and Parkinson's disease. And they determined, and this is correlation specifically, but they determined that people with DR4 and those without it, people with DR4 as one of their genetic alleles, had a 10% risk reduction against those who did not have it. So there was a similar protective effect for Parkinson's and for Alzheimer's disease. They looked at autopsied brains from Alzheimer's patients, and they found that those who had DR4 in their genes had fewer tau tangles. They had fewer of the fibrillary tangles that muck everything up and seem to characterize what we look at when we're looking at Alzheimer's brains. And looking even further, they think that this hints that tau is playing also a role in Parkinson's. DR4, it turns out, is part of a gene complex known as HLA. This is the human lymphocyte antigen complex. HLA is part of our immune system. HLA, every human has lots of little genes that grab onto stuff in the cells and like pop it up to the surface of the cell membrane and go, look at this, look what I found in the cell. Isn't this interesting? Does this belong? Does this not belong? And so these HLA genes, these little alleles, when they grab these things, maybe the DR4 goes, oh, I don't like this tangle. I go put it outside the cell. And then the immune cells come along and they go, oh, that's not a good, I don't like a tangle. And then they eat the tangle and they go, no, let's apoptose that cell. And then you don't have the tangles building up. You don't have the problems that are happening. So it's a really interesting finding that potentially could lead to more research into these HLA genes and how they work with the immune system and autoimmune disease. So if you have a gene that's not showing up the tau tangles, if you don't have that DR4 and your HLA is like, meh, it's fine. I don't think that's interesting. Then your immune system never knows it needs to take a cell out of the running. And so you can end up with cells that cause problems and then lead to inflammation that furthers autoimmune disease and can have all sorts of downstream effects. So the researchers at Stanford, with their paper that they published, they think that DR4 is involved in protective autoimmunity. It knows how to grab onto the right tau protein and to show it off and say, hey, you're a problem. I don't like you. This isn't good. And they did find in a dish, they tested in a dish, a bunch of these peptides and molecules and DR4, and they found that DR4 really, really liked a very particular protein, a peptide, little tiny chunk of a protein called PHF6. And it just so happens that PHF6 is also a peptide that gets altered in Alzheimer's brains. So DR4 is like really sticky for this messed up peptide that possibly leads to clumping and the neurofibrillary tangles that lead to clumping and neural problems in Alzheimer's and possibly Parkinson's. From this study, they're suggesting that people that they could maybe even develop a vaccine based on this.
Yeah.
Yes.
So I'm suspect.
Yes.
Of everything. When it comes to the immune system.
Oh, you
so
should
people
be
with the gene,
complicated.
With the gene, higher incidence of multiple sclerosis and rheumatoid arthritis. This is the high wire act of our immune system. So, for instance, is the CD4 T cell genes or T cells. They get recruited when cells are doing bad things, and they are involved in apoptosis. They're T cells that will go after our own cells and kill them off. Mm-hmm. When they go bad. When somebody says exactly the cells
when
like,
good cells
hey, what do you
bad.
think of this? And they're like, I don't like it, and they will attack the whole cell. So this is also a problem for people who are trying to target tumors. Because the tumor cells might be like, oh, no, we don't want too much of these cell killers here. So they go, aha, we'll knock down that system. Then what happens? Then there's over-recruitment, and these things are killing other cells. So, for instance, high CD4 T cells are now associated with the most severe COVID. Right. Because when these cells get recruited, they tend to do a lot of damage to lung and liver, kidney, that sort of thing. But then if you don't have an abundant recruitment, or if you have too good of an inhibitory regulation of them that doesn't allow very many of them to interact with cells, then you get more likely cancer. This is the problem with anything. And when you're talking about, oh, we need to go in and fix this problem in the immune system for this disease, yeah, you can cure probably any disease by fixing the immune system, but you're probably going to cause a different disease.
It's possible. Our immune system is really like 3 different systems that have been layered on top of each other and work together. Yeah, it's a very complex system.
It's not ever going to be as easy as tackling one problem. You have to take into account all of the upstream, downstream effects of the thing you're doing, because they're going to affect other areas maybe far away from the thing that you've targeted. So I'm just saying that the immune system scares me.
We should mess with it, but not too much.
Yeah,
you can mess with it just a little bit, but not too much. It's okay.
Because you got the viral researchers looking at COVID. It's like, oh, well, we need a vaccine that can prevent these CD4 T cells from being overexpressed. And then you have the cancer researchers like, no, we need to increase them so that they can take down the tumors that they're being recruited to take down. And it's almost like you've got different people trying to cure different diseases in complete opposition about what should be done to perfect the human body. But of course,
but it's not necessarily perfecting. It's not necessarily perfecting the human body. It's cost benefit. If you're against Alzheimer's or cancer, maybe this choice and treatment that we have right now is the best thing to do at this point in time. Maybe one thing at this point, that's where we are. Maybe someday nanobots will fix us all.
I'm still trying to achieve the perfect body. I don't know.
You pumping iron, or are you feeling a little fatigued?
Doing nothing.
Nothing.
Oh, I'm doing nothing. Do you know why? Because I am just chronically fatigued all the time now.
However, you're a parent.
It's because I have a little virus that's growing in the house. It's getting bigger every day. Mm-hmm. Getting into more and more things, climbing on stuff now. My goodness.
Oh, this
is an interesting article. Where? Oh gosh. I don't know where this is. Public Library of Science.
Awesome.
pathogens. Close pathogens, this is Professor Maureen R. Hansen, Department of Molecular Biology and Genetics, Cornell University, took a look at the history, the correlation between what is typically now called chronic fatigue syndrome, but used to have a much more sciencey name, myalgic encephalitis.
Encephalomyelitis. Yes, myalgic encephalomyelitis or chronic fatigue syndrome. Yes.
Yeah. So it got turned into different name. Anyway, chronic fatigue syndrome. I'll call it that because I can't pronounce the other one.
Or you could just say MECFS.
MECFS. There we go. So this is one of these diseases that everybody has been faking for the longest time. Something they estimate around 60 million or more people have been faking this disease, that they're just tired all the time when they're really lazy. Well, no. People have this chronic fatigue. However, there's no diagnostic. There's a diagnostic that varies, but there's no treatment.
Yeah.
And most doctors don't have a way of a clinical test that identifies for sure, yes, you have it, and because of this, and it's affecting here. They don't really know the cause. They don't know the mechanics behind it. And so what happens is doctors say, well, okay, let's try. You should get plenty of rest, eat healthy, and have regular exercise, like go for a walk.
Yeah, that's going to make you too tired.
Yeah. Right, because one of the things is people have unrestful sleep. So whether it's 8 hours or 4 hours, they rest as though they've gotten maybe 2.
Mm-hmm.
Even sometimes light, like going for that regular exercise walk, can actually drain them to the point of being unfunctional. And it's a real disease that's only ever sort of lost in the shuffle because doctors don't know how to define it or treat it. And so
I love though that it's like COVID-19 really brought it to the forefront because of the number of people who with long COVID, these were the symptoms. This is what a lot of people who really have the long COVID that really is lasting. This is what they have. And I remember back in the very early days of this show talking about chronic fatigue syndrome and the whether or not it was real, and scientists debating whether or not it was real. What's the cause? What's making this, what's causing it? And people thinking, trying to get away from electric fields and their microwaves and other things. I don't know what's making me feel like this. I just don't feel good. So I have to do something. And nobody believes
if you tell me it's the sound of a noisy refrigerator, then I would believe you, because that
it'll keep you awake at night.
That'll wear you out. Mm-hmm. So Hansen, though, has sort of been, I think, paying attention to this pre-COVID. And so what she's done is pretty interesting. She's collected cases of MECFS outbreaks, and where multiple larger groups of people have all sort of been diagnosed with it at the same time, and these are correlated with viral infections. What she's determined is tough is she's finding a strong correlation between MECFSs and enteroviruses, the enterovirus family.
Yeah.
Some of that's Epstein-Barr virus, that's human herpes viruses, plus others. And it's pointing out, like, the difficulty here is it seems to come about from a post-viral infection reaction. So you get the virus, you're sick, you're blowing your nose, whatever happens, you got the cold. And then you recover, and then maybe months later, the fatigue will kick in. Maybe that's when it's happening. So you go to the doctor, and the doctor does a blood test.
But they don't put things together.
There's nothing there. The virus is cleared. So it's hard to even connect it to whatever, because they can go and look at your antibodies and say like, oh, here's a list of maybe viruses that you've been infected with, and this is a deep drill down that your local doctor is not doing. They can say, oh, here's all the viruses that you have antibody reactions to, so it might have been one of those, but they don't know what you just had, especially if it was a mild and you just kind of got through it. But then the fatigue is the intense part that comes later. So just exactly what you're saying, in the global experiment that is COVID-19, SARS-CoV-2, we've seen a large number of post-viral infections effects for this chronic fatigue. And so Professor Hansen's in this paper that she's writing up is kind of pointing out that, okay, welcome aboard.
Welcome. You finally come along. Okay, great.
we have record number of people being diagnosed with chronic fatigue syndrome. However, there are already that many people and more who have it. And they didn't get it from SARS-CoV-2. Mm-hmm. So
yeah, not the only one. Yeah.
So there's other viruses, and if you create a treatment, if they do develop a treatment, which hopefully they do, that works for people who are have been post-COVID infection, it might not work for the others. If we're talking about a viral reservoir or something of this nature that's different than the COVID, it may not affect. Unless you're just talking about attacking symptoms. But if you're actually talking about clearing, finding cause, may not affect. So anyway, she's like, we need to research where it was before as well as this new direction of COVID. But yeah, there's nothing like a global experiment of getting everybody sick at once to illustrate that chronic fatigue syndrome, not only is it real, but it is virally connected. Because the list of things, because doctors couldn't easily connect it to a viral infection, lists everything from environment to gut microbiota to stress to like, there's a whole list of pathogens they think might be related to it. But the chance of what Professor Hansen is saying is like, no, there's probably a few viruses that are responsible for all of them. They're common enough that a lot of people have come in contact with it. And so we need to focus on causatives, which COVID-19 has maybe highlighted as well.
Right. Yeah. What is happening with the immune system interactions? What is leading to this ongoing inflammation, the fatigue, all the stuff that is related to it? I don't know. More research, because who likes being tired? I mean, I love sleeping, but I don't want to be, no. And the fatigue that hits people is debilitating. It can be debilitating. Another incredible science communicator out there, Diana Cowern, the physics girl, she is suffering from ME/CFS related to COVID infection, long COVID, post-COVID syndrome. And it's months now, and she is still dealing with it. And it's not easy. So, yeah, she's just one, and there are many, many others out there.
Well, so, and this is just from Hansen's write-up, but in hers, she says they're estimating about 65 million people have it post COVID, and that as many people had it from other viruses before.
Jeez, but we ignored them, or
so 100. Then what is that? 100. My math is bad at math. 120 to 130 million ish people with chronic fatigue syndrome, and we're like, well, get plenty of rest. Like, I get that the doctors always mean well and that they're only working with what they can work with, but gosh, that's got to be frustrating. It's got to be. It's sort of like how we were talking about a while ago, how there's a genetic component that takes place with yeast infections that has nothing to do with hygiene. So doctors are like, "Oh, let me teach you how to
take care of
have
yourself.
proper hygiene." Like, oh gosh, I think I've been doing that right. Or, oh gosh, I think I've been going to bed at a reasonable time. I eat vegetables, and it's still a problem. So,
eat your vegetables. Take care of yourself. And then good luck, everybody. We talk a lot on this show about air quality, and more studies over the last decade or so have reported on the links between air pollution, specifically fine particulate matter, and illness in society. And so more and more often we're like, oh, smog, or air, people who work at the little toll booths on freeways, as if they're in danger. Fire, it's bad. Volcanoes, that's also bad. But fine particulate matter, really, really bad for causing illnesses related to asthma, bronchitis, cardiopulmonary issues. So I was excited about this particular study, and I'd love to see more about it. But out of Washington University in St. Louis, these researchers looked at particulate matter 2.5, so PM 2.5. This 2.5 micron size and smaller is particularly associated with premature death. And so these researchers specifically looked at these little tiny bits of fine particulate matter in the skies. Originally from 1998 to about 2011, and then from 2011 to 2019. And they determined that it hit a peak in 2011. So fine particulate matter was going up, and we weren't really paying attention. It's like, oh, the health issues, blah, blah, blah. Now we're really talking about the health issues. But really, there is a reduction. And there has been a reduction from 2011 to 2019 in the amount of fine particulate matter that is actually in the air, which I find like, hey, maybe we're doing something a little right. Maybe we're talking about things in the right way. Maybe technology is coming along and we're not having as many coal-powered power plants, or maybe there are other local contributions that are changing things. They specifically say that this reduction is driven by exposure reduction in China, and also reduced growth in different areas of the world. So it's every once in a while it's we talk doom and gloom and pollution and trouble and world's ending, and but it's kind of nice to see that maybe we're starting to do something right, that maybe our technology is starting to come along, and not just related to the number of people on the planet, which is just going up, but also the way that we're able to mitigate some of the changes that we make.
That's one way of looking at it. When I'm looking at this map, you know what I'm seeing?
What are you seeing?
I'm seeing that. I don't know how that's possible. I just don't understand how it would be possible. Like if this was just a map of the United States and Europe, I guess it would make sense. But if you're saying particulate matter has gone down in developing nations,
well, maybe it's not growing as much. The growth of particulate matter.
Okay. I think it's a misleading infographic the way that they.
Yeah, well, so what they've said is what they looked at, their endpoint for their data, global population weighted 2.5 micron particulate matter exposure related to population levels and pollution levels over the period of time. So tied to population, per population, and pollution, people are getting less pollution that is dangerous to their lifespan.
Wait, per population?
Oh, yeah. Based on population size. Specifically, they say it's rigorous air quality management in China since about 2013.
Okay.
So China, we want to help. We want to continue to help countries that are developing nations to become better stewards of their resources and people, and help us. If we can create better ways to create cleaner energy faster for everybody, it's going to be a lot better for the people who are on the planet. But anyway, good news, everybody. Little less pollution. Even though there's more people on the planet. I'm not going to talk about those numbers anymore.
Like, if they're dividing the amount of pollution by there being more people, then I'm like, okay, I see how you got there. But if they're saying people are less exposed to pollution, I mean, we've had like, certainly automakers have done a better job of trying to maintain, keep the pollution in the car out of the exhaust. But
yes, yeah. So automobiles.
But there's a lot more cars than there used to be.
China is reducing the number of coal-fired power plants and going more nuclear. So there's a lot more of that going on. There are changes in the way that things work, but we have to balance that with. I mean, who knows where it's going to go? Since 2019, the number of Northern Hemisphere fires has increased. The number of, we've got big climate problems that could potentially impact this a lot that aren't related to mitigation effects of our own technologies and air quality. So
that's wild.
But a little good news. 2019, that was good.
I'm like, I don't know if I can handle it. I had any good news. I don't even know what it looks like.
I know you're not accepting this good news very well. I'm trying to help out here, Justin.
Well, it looked too dramatic to me. Like, I'm trying to figure out, like, what was the revolutionary technology behind this major shift in pollution globally? And if you isolated it to a few areas, I might have been like, okay, well, they've applied a new approach. But globally, including developing nations that are burning more fossil fuels,
and like there's all sorts of sustainable infrastructure that's going in places.
But
and
it's
also,
not going in everywhere.
China hosted the Olympics for a while, so they had to turn things off to clean the air. So people.
Where's this study out of? Hang on.
Washington University in St. Louis. And this is a peer-reviewed study in Nature Communications. So you too.
That doesn't necessarily. I still want to see.
Look at this study. We'll link to it in our show notes.
There's something like they don't even guess at like the reason why. Like, oh well, maybe somebody put up a solar panel in Argentina, and therefore they've reduced
solar panel. Jeez.
Like, well, I mean, like, it just there's a lot more people. Like, even if you cut emissions in half, or whatever it would be for 20-year study, a third maybe. If you reduce emissions by 25%, there's 25% more people creating emissions. Like, I'm just like not understanding how it's possible. It's that it was a reduction.
Yeah, overall.
There's too many humans doing stuff every day for that to sort in my brain.
There's more, there are more people doing things, but we're also maybe doing things better. There's a stagnation of growth in the particulate matter attributable mortality. So fewer people were dying from the particulate matter exposure. There's really, they say globally, China was the big mitigating factor, the reduction, and that it's been due to long-term efforts and change overall. But it could change. We need to keep working so that climate change and human-based sources don't keep the number going back up again. Let's live healthier, everyone. Yeah. This is This Week in Science. Thank you for joining us. I hope that we are bringing you a little bit of good news. Science. Just breathe that in. That just smells so good. Oh, that dose of science right there. That's right. If you're enjoying the show, please share This Week in Science with a friend. And then additionally, you can help to keep TWIS going. Be one of our supporters. Head over to twis.org and click on the Patreon link. Those of you who support us at $10 and more per month will get thanked by name at the end of the show. So thank you so much for all of your support, everyone out there. We just love that you're here. Hey, everybody in the chat room and over there in the Discord. I hope you're all doing well. Let's have more science. We're going to come on back with more This Week in Science right now. I think I'm going a little batty. Just a little batty.
I'm just going to leave it there.
I'm
not even going to question it. I'm just going to...
Thanks a lot.
What? Why? Goodness, what do you mean, Kirsten?
What do I mean? Researchers out of University of Illinois Urbana-Champaign have been looking at the good and the bad of bat boxes. We love bats. Bats are pollinating species. They're wonderful nighttime species who also, they get the insects. So you have mosquitoes and those flying things that bite you in the nighttime. The mosquitoes helped get rid of the things that you probably are not excited about in the nighttime. So we want to help the bats. And so a lot of people are like, let's put bat roosts. I have a tree. I have a house. I'm going to put a bat box on my tree. I'm going to put a bat box on the house. And the question is, how good are these boxes for bats? And I personally am interested in this because I've been thinking about this. And you go online and everywhere you search, it says, make sure you put the bat box. Make sure it has like southern and eastern exposure to the sun, and that it's the bat box is like in the sun, and the bats will go in there and they like it hot. And I'm like, wait, real? And this is just general information. Apparently, this is not confirmed and is probably very bad for the bats.
Yeah.
So what do you think happens when you put a bat box in the sun in a fairly warm climate?
You cook the bats.
Yes. And that is exactly what they found. They and others have documented that different bat boxes of shapes and sizes and materials can reach really dangerous temperatures. And very often you end up with poorly planned bat boxes because you're just a person who's like, I'm going to save the bats. And you put a box on the side of your house and then the bats cook and you end up with a bunch of little tiny bat bodies piled up underneath those bat boxes on really, really hot summer days. Bad. It's bad, everybody. So the biggest concern is overheating. And they think that what's happening is that people are inadvertently creating what's called an ecological trap by putting bat boxes in places. It's something that the bats might like, but it's not necessarily the best housing for them. So the bats might inadvertently choose this bat box, but that bat box might not be where they should be because it's in a bad place for them or it's just poorly designed. And on top of overheating, the opposite of that is freezing, because if it's exposed and in the wrong place, not only do you have the problem of hot summer sun, you also have an issue with no insulation and low temperatures in the winter that can freeze bats. So the researchers are assessing these conditions and trying to figure out how to make better bat boxes for
Build a
bats.
better bat box. So
Then, but then you have to put them in the right place so that raccoons can't get them, or snakes, or other predators. And then what about ticks and mites and other ectoparasites that might really like living in those bat boxes and make it...
We're not going to solve every bat problem.
Well, these researchers publishing in Conservation Biology, they would really like us to do a good job of trying to help the bats. And I think we can do better. We can do better if we try. We could save the bats if we build better bat boxes. And I think my big take home for this is don't just accept what you see online, because you're probably using unconfirmed information to create the best.
Unless you listen to this show online, in which case, believe everything.
Believe it all.
John Morano, in a comment referring to the study I was, my comment on your study about there being 25% more people. Yeah, over the course of that 20 years, according to some website I just pulled up, 1999, we were just over 6 billion people on planet Earth.
No, right.
And then what was it? 2019 was the end of that study. 7.764 billion. So, not 25%, but maybe 20%. Actually, it would be a little over 20%.
100% of humans die eventually.
But yeah, we have a population growth rate of over 1%. Mm-hmm. So in 20 years, you would be over 20% more population. And that's going to be true of every 20 years until we run out of food, space, air, and ammunition.
So in the meantime, don't worry about us. Worry about the bats. And if you are...
Let's focus on the bats, people.
Let's focus on saving the bats. The researchers who were involved in the study at the University of Illinois, Urbana-Champaign, they have a website, which is wildlife.nres.illinois.edu/safer bat boxes. We'll link to this on our website. And they have tips for making bat boxes safer for bats. So they'll help you with where you place them, how you color them, what you might want to build your bat box with. So if you want to help with the bats, make sure you're going to a well-resourced website before just going batty like I do.
Yeah. Or if you're in an urban environment, an urbanized environment, you might want to just focus on the animals that are already there and giving them a good place to live. I don't see why people aren't making rat boxes. That's an animal.
If you build a bat box wrong, it could become a rat box.
It's an animal in your community that needs someplace cozy and warm at night. That's a little bit cool in the summer. Rat boxes, people. Why not? It's that or your house, right? That's why they keep trying to get in. They don't have any rat houses anybody built for them. Go out there and build some rat houses. Take care of your community.
That's what we're doing with the bat houses. Community bat.
Don't just be one animal specific.
All the animals. My house is home to all the animals now. Yes, okay.
Build a raccoon mansion in your backyard. Forget the tree house. Raccoon fort.
People build squirrel houses. Why not a raccoon house?
A squirrel house. Perfect idea.
I apparently only plant things in my backyard that help bunnies survive now. So that is what I'm doing. Nice. I'm helping the local bunny population. But there are lots of ancient things that have died out over the years because nobody was taking care of them. So Justin, did you want to talk about some old humans and maybe reverse some of your discussion from previous weeks?
Yeah. So first I'm going to start with the 1960s. Researchers in northern Israel excavating a 1.4 million year old Homo erectus site found hundreds of stone balls. I think they found at this one site almost 600 of these. And they were made from different types of rock, basalt, limestone, stone balls from 38 different layers. So this could have been, created over 100,000 years even. Like they're from all sorts of different eras. And they're described as plum size. Now, I don't know how big a plum grows over there. A California plum gets pretty big, but I can kind of picture this as being something that fits in the palm of a hand. So it says, these rocky orbs had no discernible purpose, and some speculated that they may have been debris produced while making other tools. Now researchers suggest that our ancient relatives intentionally crafted these spheres, perhaps for the sheer joy of creating symmetry, according to the article in Science. I'm going to just jump in right there and say no. I don't think it was from the pure joy of creating symmetry.
You don't know.
I don't know, but I'm going to speculate because I think that they were probably not used for hunting, but likely used to throw at things.
That makes a lot. I mean, looking at these spheroids, they look like something that you would throw, but maybe they were playing bocce ball or baseball.
Look, it's possible. It's possible they set up ancient bocce ball courts and that sport goes back way further. It's probably. But the fact that they had so many of them at the sort of home site makes me think that they were used to ward off things. So, you've gone there and you've done your big hunt, and now you've brought the big roast beast or unroasted beast back to camp. Now here come all the other animals who are like, oh, we want to eat your food. How do you keep them away? You either can wave a stick at them or you can throw a rock. And these are people who are making some stone tools. So, my goodness, well, boy, you throw a rock and it, sometimes it goes far and sometimes it doesn't go far and sometimes it kind of flops around. And then somebody's like, oh, I threw this rock and went right where I meant to throw it. And so they went about making more rocks like this. That's my guess. They used it to throw at things. Maybe you could hunt one of Kirsten's backyard bunnies with it. But beyond that, probably not a great hunting tool, but pretty good maybe at throwing it. I don't know what's there. Coyotes or whatever might be trying to wander into camp. Or a lion even. Like, you don't want to get close.
Saber-tooth things.
And hopefully you don't throw it at other humans. But the researchers did 3D modeling. And they could tell that these were from the scarring and little nap signatures on these, that they were intentionally crafted to take on these spherical shapes. That these aren't like river stones that were collected because that would have been too smooth. It wouldn't have had the sort of precise napping that was required to get to that point. So, and there's some point out that we don't have the full context because, of course, these are 1.4 million years old or something. So for all we know, they were painted bocce balls, like you're saying, Kiki. And this was part of some sport or some entertainment.
What do you do when you're biding your time at camp in between hunting outings, or how does it work?
Yeah.
How long have human ancestors been trying to entertain themselves?
Oh, probably
and protect themselves.
Imagine? Well, they did have the stars to watch. Those must have always been fun. Anyhow,
I wonder though. This kind of a structure, it's very, like you said, it's something that was on purpose. This is something that they possibly learned, the napping technique. Maybe this is also early learning how to do flint knapping or learning how to create structures. Maybe these were instead of teaching your child how to make a knife when
Yeah,
they might
yeah,
still
smart.
maybe you start with a ball. Maybe maybe these were maybe they had lots of different uses.
And so actually what you just point out is so smart, because one of the things that we're almost always missing from every archaeological record are children's toys.
Mm-hmm.
And yet we have all these small rattles, or like this, small balls or small artifacts that we find made, and we go, oh, yes, they were ritual use. They had some sort of because it didn't have a clear function. Chances are we're finding toys all the time, but archaeologists become archaeologists because they were not allowed to play with toys as children. So they don't recognize them.
Any archaeologists out there, feel free to pipe in on that one. But
But no, you're right. I mean, it could just be that these were I don't know, made by children, but they could have been made for children for sure. They could have been children's toys. Like, why not? Like,
if they're found around the campsites or wherever the habitation locations
yeah, don't
were.
wander off here. Throw rocks at each other. That's a
Right.
what
Or,
could
hey,
happen?
kids,
We don't know.
here, try and make these rocks and then throw them at each other. No. Throw them at the saber-toothed coyote Don that is trying to attack her.
Or that.
Yeah.
Okay. All right. So then this brings me to, I'll finish up with so 2 weeks ago, I said, oh, I don't believe in Denisovans anymore. I think those were Homo erectus Neanderthal hybrids, all of them.
Right.
I spent like the last week or so thinking about it, and I think that's completely wrong.
Okay. Why so?
Well, because it doesn't add up to... It still has all of the problems to solve of these hybrid populations being in the exact right area, common human ancestors moving through and interacting with Neanderthals and Denisovans, or this hybrid group of them. Mm-hmm. Okay, so the story so far, Neanderthals develop out of Africa, and around 700,000-ish, 600, 700,000 years ago, diverge from Denisovans. Right? They split paths. One goes into Asia, one goes into Europe. And there's always being mentioned a mystery archaic DNA somewhere in there, but we'll just ignore that because we don't know what that is. So Neanderthals to the west in Europe, Denisovans move east into Asia. And then, about 600,000-700,000 years after they have split, it looks like they meet again at Denisova Cave. And they find this bone, and they do the DNA, and they find, aha, we've discovered a Neanderthal with some unknown DNA that we now can tell was connected to Neanderthals about 600,000-700,000 years ago. We have a hybrid between the two. This is the amazing discovery at Denisova Cave. Okay. Here's the problem I'm finding, is the order of discovery has dictated everything. Okay, so Kiki, you and I are going to go and we're going to sequence the genome of a donkey.
Yes.
Okay?
Okay.
And so we find an old donkey skull in the desert, and we find out we can extract some DNA from it, and we do. And now we have the genome, for the first time ever, of a donkey.
Yes.
And then we go and we find a horse, and we sequence the genome of the horse. And we find that the horse actually is half donkey and half archaic ancestor to the donkey that was split by a long period of evolution. Well, what turns out is our hybrid that we think this horse is actually a horse. And what we first sequenced and called donkey was a mule.
Oh, that's sneaky.
Sneaky, right? So, under this new scenario that I'm looking at, what would happen is around 700,000 years ago, an ancestor, common ancestor of modern humans and Neanderthals, is in Asia, Western Asia, likely. It runs into Homo erectus. They mix. Then their offspring go into Europe. Hundreds of thousands of years pass. Somebody finds a couple of Neanderthal, some skulls, and we sequence it, and we go, aha, Neanderthal. And so we have a Neanderthal genome.
Yes.
It's 100% classic Neanderthal genome, because that's all we know. Then, what is it going to look like if we ever find a Homo erectus genome? It's going to look like it's half Neanderthal.
Right, that it's got some shared stuff in there.
It's going to look like it's half Neanderthal and half something that was connected to Neanderthals about 700,000 years ago.
Right.
That's maybe why all the Denisovan DNA that we're finding looks like it's half Neanderthal, because in our description of Neanderthal, we have human common ancestor and Homo erectus. Now, I thought of that and I went, well, gosh, that's kind of interesting, but what about all then the Neanderthal DNA, which is actually higher in East Asia than it is in Europe? Oh, wait a second. That's also where all the Denisovan DNA is. Wait a second. What if what we're calling Neanderthal in that East Asian populations isn't Neanderthal, because our Neanderthal is defined in this scenario by Homo erectus. And
right.
That means, wait for it, it's possible that human ancestors, the current modern humans, 50 to 60, 70, 80,000 years ago, when traveling east, actually encountered Homo erectus and interbred. Direct. No little buffer zone, friend zone with a Denisovan.
Right. So it could have been, modern human and Homo erectus. And
because it also would explain why on remote island populations in Indonesia and in the Philippines, the indigenous people have the highest rate of Denisovan DNA, despite no Denisovan or Neanderthal fossils in the area, but an abundance of late existing Homo erectus, including the Homo floresiensis. So it makes it so much easier, because you no longer need all these intermiddle populations or Neanderthal and then a reintroduction with the Denisovans later. If humans just go direct to Asia and then mix with, if Denisovan is actually Homo erectus. Anyway, my point is, this is my new prediction. Modern humans mixed directly with Homo erectus in East Asia. Neanderthal is a mix of the human shared human ancestor and Homo erectus in all of Europe, and that every Denisovan DNA that we find is always going to be a hybrid with Neanderthal, because half of Neanderthal
is shared with modern human
is shared
already.
with Denisovan, because I think Denisovan DNA is Homo erectus DNA. We'll never find Homo erectus DNA anywhere until we disentangle.
That's a fascinating idea, and we're not going to get Homo erectus DNA directly going that far back, because DNA doesn't, as far as we are able to do, get it to
unless we already have it.
Preserve it that much, unless
we already
in the
have
genome. Yeah, interesting.
Well, no, but in the genome, but also like one of the things is there are estimates of Homo erectus surviving much later then people think of Homo erectus as 2 million years ago.
But it could have been longer.
That's when it starts. Yeah. But the end date of Homo erectus is still open, with some saying as recently as 30,000 years, some more in the, like the fossils we have found.
Which is a real overlap with modern humans.
Major overlap,
Neanderthals and everyone, yeah.
Yeah, and I'm also suggesting, I don't think that there is Neanderthals in the Far East. I don't think that's Neanderthal at all. I think that DNA is, what we're describing is just Homo erectus. And when you say Homo erectus too, it's going to be a much more diverse population of Homo erectus than there are of pretty much anything else, because it's been existing for as long. That's how you, like Homo floresiensis, you could call Homo erectus. It's islandized, it's morphed.
Yeah. I think, I mean, this is definitely the kind of conversation that needs to happen with somebody who does these genomics, who does the phylogenetic analysis of human DNA, of these, looking at the human ancestors going way, way back. I mean, this is a puzzle. I mean, I think the thing is that it's great to have these conversations and these open questions, because honestly, nobody really knows the answer at this point in time. Well, we're calling it the braided stream. We have a lot of data, but there's still more to learn.
If we get like a Homo floresiensis, or I keep forgetting the name of it. There's another
Heidelbergensis, or
no, there's another small person in the Indonesian, one of those islands down there. It's separate from the Homo floresiensis. But I keep forgetting its name. If we can find, and these are like possibly 18 to 30,000 years, if we get DNA from one of those and it comes back as Denisovan, I will feel like, we're pretty far along that route. So there's ways that we could solve this. What I would love, though, is I would love us to get a guest who's been studying these genomes to tell me if it's possible that Homo erectus and Neanderthal hybrid could work.
Or, what are and if not, then what are the reasons that hypothesis shouldn't work based on the current understanding or current data?
Oh yeah, but
this is all we got to look at the genetics, right?
Yeah. What I'm asking for is, get me out of this rabbit hole. Tell me why it's wrong so I can stop thinking about it. I can't do anything with this information. I need to get it expunged from my brain, so my brain stops going, wait a second, whoa, hold on.
We need to get you some help.
Well, this is the other thing, is like we were talking about looking at the pluripotent cells for signs of early pregnancy termination. Miscarriage.
Yeah.
Those genes in the Western world are strongly correlated. They're associated with Neanderthal inheritance.
Right. Some of them, yeah.
A lot of it is. It's heavily influenced by Neanderthal ancestry. So the thing is, why would, and there's been some speculation, Neanderthals, because they were hunter-gatherers, they didn't want to have large populations evolutionarily. It was to their benefit to have less children. I think that's nonsense. I think what happens is that because of their hybridization event from a much different part of humanity and this early Homo erectus, Justin theory, that didn't make them infertile, but made them less fertile, made it more difficult, complicated, and actually likely made many of them infertile.
Hybrids are very often infertile or have fertility issues. There's documented impacts of hybridization of various species that result in infertilities for various reasons. But yeah, that's fascinating. Laura D is asking in the chat room if we could interbreed and produce fertile offspring. Were we different species? And that's a really interesting question, because you have the biologists who are the lumpers and put everyone together, and then you have the splitters who split everyone apart. And even in talking about bird species or animal species, like Patrick Pecoraro was saying, and you could have an animal that's the same species-ish, but it's separated geographically where they normally don't come together. They're geographically isolated, so we call them different species. But they can hybridize sometimes and come together because of geographic incidents, and they do, or they mate just fine. And it was just a geographic isolation. Sometimes it is actually genetic or other anatomical factors that are involved in actually blocking the fertility aspects. But our concept of a species is, I'm throwing things around here. Our concept of a species is just a very fuzzy wuzzy issue. We have some that we can say, yes, they don't interact. We don't interbreed, but there are others that are just on that line, which is very interesting. So possible.
So bears can, from different regions, they can interbreed. You can get a brown bear and a polar bear, and they can have a baby. Lions and tigers can make ligers. I don't know if a liger is reproductive.
I don't know.
Horses and donkeys seem like the same thing.
Not.
And they can reproduce. A mule that then cannot reproduce after.
Exactly.
So that's the thing. There's a little bit of genomic dice roll in how the components that you inherit can operate the next time round. And with Neanderthals, what it looks like is they had some major issues with reproductive health that may not have evolved naturally. I think that's a clear sign of some
or it was a trade-off because maybe their reproductive aspects, it was a trade-off for survival in other ways because they were living in colder climates more often. There was they had slightly different adaptations to the ecology of the areas that they were in. So these are big questions. They're fascinating, but we can't answer them tonight. That's for sure.
We'll try though by continuing to talk about them until my brain is reduced
or
to a few cells in a dish.
Put you in your place. Right. Okay. Cells in a dish. Let's talk about those. We love cells in a dish.
Wait. What did you say? I was talking. You were saying something about. Anyway, yes, in the dish.
Cells in a dish. Researchers published in Nature Communications last week their work on this wonderful dish brain system that we've talked about previously on the show. Researchers have been trying to demonstrate synthetic biological intelligence with a neural network of cortical neurons in a dish brain that were trained to play Pong. We talked about this, and so the brain, these little cells were able to go, oh, I'm connected to a monitor, or this is the feedback that they were getting, and they were able to, cells in dish, able to play Pong with a computer interface. I don't think they knew they were playing Pong, but they learned based on certain feedback how to react and how to turn themselves on and turn themselves off, stimulate or reduce their output. Now, these researchers just publishing in Nature Communications, their study critical dynamics arise during structured information presentation within embodied in vitro neuronal networks. So what does this mean? It means that the researchers have taken their dish brain and they put the dish brain to the test against hypotheses about how the brain works and how neurons in a neural network work together. And because of what they had been able to do, teaching the neurons to play the game of Pong, they were able to measure what's called criticality of clusters of neurons, on how clusters of neurons interacted and reacted to feedback that was presented to them. And they discovered that they needed feedback responses. So feedback loops led to the real activity and real learning of the network. And there's been a hypothesis in neuroscience for a while that's called criticality event. leading to a neural network choosing one path of action or another, being active or not being active. How is it going to be going to respond? And based on what they saw in their dish brain, they were able to show that this criticality hypothesis was supported. So the brains needed to have feedback. And when they had feedback, certain populations of the neurons went into what was called an avalanche mode. They all decided to work together. It's like we have a threshold, a potential that leads to a single neuron firing or not firing. But in a neural network, there's also kind of a threshold. And these researchers thinking about this, the importance of this is that sometimes when neurons all go too much, then you have things like epilepsy. You have storms in the brain. You have migraines. You have things going wrong because the neurons aren't working together in the proper way. They're not acting on feedback correctly. The opposite end of that is a coma, where the neurons are just not reacting to feedback at all. And so in this gameplay simulation and stimulation, they were able to show that this neural criticality is just a base feature of how neurons interact, and it's really important for neural networks to move going forward. Learning, feedback, and there has to be a reorganization of the neurons working together to get to this critical avalanche state where they go, boom, we're firing together, everybody. Let's do it. We're a team. And Daniel Smith in chat room, yes, the old adage, neurons that fire together wire together. So this is the, why would a group of neurons fire together? This critical dynamics, this criticality is part. It's not sufficient to make it all go, but with reinforcement, neuronal networks get to this this criticality, which allows an avalanche of activity within the entire neural network. So why is this important? This is important to understand cognition and how our brains work generally, how we can potentially move forward in understanding disorders like comas or epilepsy, and also how we can create better brain dishes, dish brains, video game playing brains in a dish. All better.
At least we've given them something to do in there. We were talking about the Homo erectus getting bored. Boy, being a brain in a dish.
Brain. Ugh. And then, what is it? What was the brain? That was the Steve Martin movie, where they're brains in jars. Blair's favorite movie. Was it The Jerk? Was that the one where he ended up in the brain full of jars? The jars in the room full of brains. Anyway, there was
vague recollection of something you're describing there.
A study this last week that people were talking about a lot online. It was published in the Journal of Pediatrics. Researchers showed that when you put complex breast milk components into infant formula, that the babies for 12 months, if they were supplemented for 12 months, it raised their IQ by 5.5 years of age by 5 points. So complex breast milk proteins like lactoferrin, MFGM, others, these supplements led to increased IQ. But this is specifically for infants. And I just wanted to bring this study up because...
Infant IQ?
5.5 years. So child IQ, they measured the IQ at the age of 5.5. Complex skills, executive function, learning, inhibition, those kinds of things. The Man with Two Brains. Thank you very much. That was the movie. Yes, Paul Disney, you guys got it. The Man with Two Brains. That was the one. But the coverage of this paper out of the University of Kansas has been bothering me because it makes it sound like, if you give this souped up infant formula to your babies, you're going to raise their IQ. But they're not
it sounds
saying it's better
ridiculous.
than breastfeeding. This is because a large number of infants are not breastfed for various reasons. There are, all sorts of reasons why breastfeeding doesn't pan out for people. We're not going to go into that. And it's been a question as to how can we make formula better. So this isn't a suddenly infant formula is better than it is.
After a hard day's work, there's nothing that rejuvenates me better than a hearty cup of breast milk. Why, I just love it.
No. Yeah. So if babies are fed formula that have more complex profiles for carbohydrates, fats, all those things, and are closer to that of natural breast milk, it does help with cognitive development, but this is not an either or. This is just helping to make breastfeeding formula or baby feeding formulas better. Yes? Yes.
But I need that extra edge throughout the day. A little bit of breast milk in my coffee. That's what I go with.
Soup up your brain skills.
It also does wonders for the luster of my hair.
No, don't. Gosh. Oh, the internet. My last study for the night. Let's not stay up too late because yet another study has shown that when you deprive mice of sleep, it causes neuronal cell death in the hippocampus. They didn't look at people, but because they kept mice up for 2 days and didn't let them sleep, they're able to say this is bad. Sleep deprivation. This is a study published in the Journal of Proteome Research in mice. They determined and they were able to correlate the sleep deprivation led to neurological damage in the hippocampus. In these mice, they looked at the hippocampi and looked at the proteins that were involved, the genes that were involved, and what was actually happening with the mice. The mice also really got worse at navigating a maze over the couple of days that they weren't sleeping very well. Sleep deprivation affected their behavior and cognitive abilities, but they determined that there is a protein that's called pleiotrophin, and it declined in sleep-deprived mice. And they determined that this loss of pleiotrophin might be involved in a molecular pathway that leads to hippocampal cell death. Pleiotrophin is also implicated in Alzheimer's and other neurodegenerative diseases in humans. So the big thing here is that pleiotrophin, it decreases, and when it decreases, it affects a whole signaling cascade inside your neurons that actually leads to apoptosis of the neurons. The neurons go, I'm not getting the protein kinases that I'm supposed to and the things that aren't happening, and we should kill ourselves. So they saw that in the mouse hippocampus. They did not see it in human hippocampus because that's not what they looked at, but they're going to potentially be looking at it as a novel biomarker for insomnia-induced cognitive impairment. So maybe it'll help us in the future. In the meantime, go to sleep if you can.
I wonder, is that like an attempt by the brain to fix something?
Probably. This is
like you're not sleeping. Why are you not sleeping? Oh, it's probably the hippocampus. No, it would be the amygdala, wouldn't it? That's usually which is the one
kind of in the limbic system.
Which is the one that's always sending out the like, danger, danger, danger.
That would be the amygdala, fear, anger. Okay. Yeah, amygdala. Yeah. Hippocampus is more memory, so
it's gonna be. You don't want to lose your memories unless that's what's keeping you up at night. And then see, maybe the brain's like, okay, we're having a problem. We need to delete some files. It's what we need to do.
Right.
I don't know if the hard drive is full or not, but this is a step I always take. We're going to delete a bunch of files, and then we're going to shut down and restart.
So that's that's the question. Why would a reduction in this particular protein impact such a cascade in the cells, lead to cognitive, like, basically, why is it like, hey, you're not sleeping, so we're gonna, maybe it's because it's resource management. We gotta get rid of things because we're not functioning properly. You gotta slim down so we can sleep better and put energy where it needs to be put, because sleep, as we know, is also kind of a brain energy management type. It's when the brain clears things out, and there's a lot of body functions related to metabolism during sleep.
No, I love the idea that the body has a regulatory system that's like, okay, the brain's been doing too much thinking.
Shush.
We need to just, shush. We need to shush the brain. It's probably found out, that the sun is a nuclear fireball, or that it's,
a massive incandescent plasma. Yeah.
It's learned too much. That's what we know. We don't care what it learned. We don't know what the information is. We're just going to start hitting delete buttons. Maybe we'll get lucky.
Yeah, but if you don't want to have the delete buttons, and if you can get your sleep, try and get some sleep. It's probably better for your neurons if you're a mouse. We don't know for sure in humans because we haven't done that study, except maybe because it might be linked to Alzheimer's and other issues. So I think that what I'm trying to say is that we've made it to the end of the show. Have we done it?
Oh, I think we have. Look at that. A tight 90.
Oh, close-ish.
Well,
yes.
Once it's edited.
Yes, absolutely. I want to say thank you to everyone for joining us for the show tonight. Thank you, all of you in the chat room, being here chatting while we are doing the show. Thank you, all of you in the Discord who are talking. It's great. Ooh, Arnlore has taken a clip over on Twitch. The moment that Justin forgot was on the internet. Okay, I got to look at that one later. That's going to be fun. Thank you, Fada, for all your help with show notes, social media. It's really, I can't do it without you. Gord, thank you for Arnlore. Thank you for helping with the chat rooms, making sure everybody's happy and nice in there. Identity 4, thank you for recording the shows. And Rachel, thank you, as always, for editing the shows. And of course, I cannot go on without thanking our Patreon sponsors who allow us to definitely can continue this show. Thank you to Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, Rick Loveman, George Corris, P.L. Velazar, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Jeffstad, Jonathan Stiles, a.k.a. Don Stylo, Ali Coffin, Reagan, Shoe Brew, Sarah Forfar, Don Mundus, P.I.G., Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie Bahal, D. Disney, David Simmerly, Patrick Percoraro, and Tony Steele. Thank you for all of your support on Patreon. And if any of you are looking forward to having me read your name at the end of the show, or just want to help the show out, head over to TWIS.org and click on the Patreon link to help become one of our Patreon supporters. On next week's show,
we will be back broadcasting live from our YouTube and Facebook channels, 8 p.m. Pacific time on Wednesdays, and then we do a second show, 5 a.m. on Thursday, Central European time.
Same show. Want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found, and if you like the show, get your friends to subscribe too. That'd be great.
For more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twis.org, and you can sign up for a newsletter if you like.
We might send one someday. You can contact us directly. Email me, Kirsten at thisweekinscience.com, Justin at TWISMinion@gmail.com, Blair, and congratulate her on her little bambino at blairbaz@twist.org. Make sure you put TWIS somewhere into the subject line so your email doesn't get spam filtered into addition of brain playing Pong and then end up with a old rock thrown at it by some kid who lived in a cave. We'll never get it. Never, ever, ever.
And we want to get it. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
And we will be back here again next week, and we hope that you will join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan.
and we're back.
La 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 la. I got a plan, and you know
More
the
This Week
after
in
show.
Science. Second half. Oh, it's the after show. Oh, we're further along than I thought.
What?
Yes, Arlen, you're the only one to ever send in a haiku. I should stop asking, because I don't think people know how to write haikus.
I think there's been more than one, haven't
There's
there?
been a couple. Yeah, just a couple. But I think it was Arlen or every time.
Same one every time. That's always. What is this clip? What is this saying? Oh, it's a wonderful clip. That's going to be great. Thank you, Aaron Lore.
Good
Love
grief.
it.
What is it? What is it?
Oh, it's your wonderful advertisement for breast milk for
Breast milk.
breast milk for the after work cocktail hour. Oh, jeez.
Well, you
Aaron
know,
Lore, that's ridiculous. Over on Twitch, I do
I think
hope
that's fantastic. I think I
would
fully
love to see
endorse
more
the use of that for selling the promoting of breast milk drinking as an after work cocktail.
no. It's
Fully
fascinating though. You go on the
Haven't
internet.
seen it, but I
though.
fully endorse it.
You go on the internet.
Don't know
though,
what it's
and-
about, but I completely put everything of my trust behind it.
I don't know about you, but you go on the internets and the advertisements. There's some company like for some reason they want to sell me this colostrum shake, and that's basically the early breast milk. But it's fascinating. I guess that's a whole thing. For better skin, for better hair, for better digestion, drink colostrum, chocolate flavored. Seriously, my Facebook ads. I don't know why I deserve it. I don't know why. It's either Temu or that. Hi, Cappy. Cappy knows. Cappy knows this show's over. Meow, meow. She's walking around down under here saying, meow, 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 meow. You got lots of things to say tonight. Oh, and the purrs. She's purring too. I don't know. I'm happy you can be here again, Arn Lore. Yeah, I'm glad you're having a good day. Concussions are awful. fatigue syndromes are awful. There's so many things. Your brain can be impacted. Your body can be impacted, and yeah, no fun. So those of you who are here and able to be here, I appreciate it every time. And Vada, it's time for you to go watch the Ashoka episode, huh? The character that wasn't in the canon until George Lucas said it was. Anyway, Peppy, hi, meow, meow. She needs to say hello to everybody because she's got to. She's a purse. Ugh, Aaron, Laura, that's awful. I'm sorry. Everyone, day by day. Yeah, it was Kitty. That's Cappy. That's Cappy. She's gotten both of my cats recently, very vocal. They talk to me all the time. Well, they've always been vocal, but even more so. Yeah. What you got going on there? I lost Justin. I don't know where he's gone. I am tired too, Fada. Oh, there he is. And now my other cat, Stella, is meowing from upstairs somewhere. They have so many things to say. R-in-law. It's possible. Who knows? I don't judge. No judgment here. Yeah, I hope everyone who is, I hope maybe we're helping people get through their exodus from Burning Man. Maybe we're helping helping you get through the morning, get through the night, wherever with This Week in Science. Not Burning Man, Muddy Man. Are you muted, Justin? No. Yes. Yes, you're muted.
I'm so sorry. I did not.
There you are.
That.
Yeah, I was like, what are you doing? Oh, I'm just going to talk to myself. That's all. And everyone who's still here.
I can't bring up the thing that I wanted to share.
Do I want to know?
Okay. It won't let me. Oh, it's because it's an interactive piece. I have to find the website. Oh well.
Oh. This is a big Yellow Jacket year, Shubrew. Yes. Lots of Yellow Jackets. Agreed. I think I saw something in the news about that, actually. Maybe I didn't. Maybe I made it up. No, Marin County, a month ago, Yellow Jacket attack spike. Worst I've seen. Aggressive singing Yellow Jackets buzzing in the North Bay. Another Oregon Willamette Week. Are there fewer bees and more wasps this year? Interesting. CBC calls to remove wasps' nests are surging.
Uh-oh.
Interesting. That's fascinating. So there's a bunch of this is north. Oh, you're in NorCal. Yeah. Corroborating reports in the news, but I don't know if anyone's done the data there, but yay, Yellow Jackets. I don't like Yellow Jackets. I like bees. I like things that are good for the flowers and the trees. I'm sure Yellow Jackets are in there too somewhere. They're in the ecosystem. They're important. I just don't like them. The stingings. I'm tired.
Oh no.
It's a tired week this week.
I don't know how to share. Oh, present.
Yeah.
All right.
What do you
want
want?
to show that thing that I said I wasn't going to show.
What is it?
The thing related to the story I was telling, or should I skip it?
Is it a map?
Yeah.
You really want to talk about this, don't you?
Well, kinda. And I kind of don't, but it's once you see it, you can't unsee it.
Sure. I'm not in charge. Is this wait, it's nothing indecent though, right?
No, of course not. Okay. So I see this. Okay. I have to what do I oh yeah. Hang on.
You can always send me a link in the private chat and I'll share it. Or you could just share a link.
Yeah, the problem is yeah, sharing is there we go. No. It's not
Technology
like so
foils Justin again.
Yeah, I got a picture up here. I'm just gonna do this. I'll just do it this way. This will be the easiest. Okay, hang on. All right. Am I sharing? I can. It still shows me. No. Yeah, I need to go and do all sorts of settings changes. Okay. Let me find this. Give me 1 second.
Is this something you downloaded? Is it a link?
Well, the thing is, I got pictures of the map that I downloaded, and I'm having an issue sharing that. Well, actually, I'm having an issue sharing my screen even because it wants me to go into Google settings and.
Oh no! Yeah, no, but yeah.
It's like a whole thing. So let me see if I can find the website real quick.
That would be great. I'm tired.
Cappy, hi. What do you want? You want to go to bed? You want to go sleep?
Yes. In the meantime, does anybody watch Star Trek Enterprise? And can they tell me if season 3 is just going to be the sad, dark season, the entire season? Because we're like 3 episodes into Star Trek Enterprise season 3 at home, and it's so dramatic, and everybody's weird and sad and angry. And does it get any better? Is it gonna get any better? Because I think my family might give it up. Wait, whatever Justin was doing, he is now completely gone. Oh, here he comes. Coming back. You're like,
yeah,
I can't even. I just gotta go right now.
I'm hitting all the wrong buttons. Okay, so I put link number 1 in the private chat.
Okay, I got that. Okay.
So let's pull that up.
I can help you there. Come over here. Let's go in there.
And then just a couple of these down. This isn't the original source, but it's the first one I found of it.
Wait, the
map.
Oh, wait,
just
the
that
map that's there. Yeah. That's okay. Here we go with the map. Back to the Denisovans. There we go.
Okay. So, this map.
Thank you, Gord.
Shows the distribution of Neanderthal and Denisovan, supposedly DNA through the world. Now, under Justin theory, the Denisovan and a portion of this Neanderthal would actually be Homo erectus, this ancient human form.
Uh-huh.
And you can see, based on this list, this map, this heat map, almost nothing at all. There's nothing there in Africa from these archaic forms. And we see a big hit when you get down into Oceania, the Philippines, Indonesia. It's a little higher in Southeast China, this sort of thing. Now, what's interesting is I would then conflate some of those Neanderthal findings as also being Denisovan, as being the same thing as Homo erectus, right? So the thing is, we're lacking morphological. We don't have morphological evidence of this. We don't have the skulls that they find in those places with high Denisovan are Homo erectus fossils. They're not finding these hybrids that should exist.
One tooth, or like a pinky finger.
Well, that's a finger in Siberia, but but they find there's Homo erectus skulls. Like one of the more recent ones is in Indonesia, but there's a bunch in Cambodia and other places like this. Okay. So here's the second map.
Oh, you've given me another thing. Okay, so I'm going to stop
Right.
sharing
So
this
look
one.
at this map, and also notice that over in Venezuela, Ecuador, the top of South America, it's suddenly a very low Neanderthal count. It's really interesting. It's like lower Neanderthal than a lot of places in the world. And that just northern edge of South America.
Okay, so you just want us to...
Flip to the other map. This map, you notice we've got the top of North America is dark. Africa is pretty dark there. And then we get highlights in Southeast Asia and in Oceania. Now this heat map is of global penis size. And when I'm looking at it, it seems to correlate pretty strongly with the first map. Now, part of the difference in these maps is one is by country, and it is the entire populations. And the other one is more looking at where they have found instances in indigenous. So there is a little bit of, these are crude maps to begin with, but they're also not looking at quite the same populations. But the maps indicate to me that a morphological difference may have existed between the archaic human and the modern humans, which would actually fit a couple of things. One, current modern human survival strategy has been heavily breeding. We are more into sex, I think, than the archaics. I think they would have been, if you took an archaic human and put them on the planet today, they'd use the internet to go to Hunt Hub to look at hunting videos all day, because that's what they were really into. The other thing is, we've talked about before how Neanderthals develop very quickly from childhood to adulthood. That means less time in puberty, which means less hormonal exposure, which might also mean you could also have less androgen receptors. That whole formative, sexual organ formative phase that current modern humans go through would have been shortened, which would also give you this result. So it could also just be a shorter maturity, a quicker maturity, accelerated maturity phase could result in this in archaics.
I think it's an interesting question, but I mean, why not? What genetics are involved, and how did it get here?
And I think it might be more
That's
hormonal
interesting.
than direct genetics, but I think it
Yes,
would be
absolutely.
an interesting thing to pursue, to look at something like androgen receptors in archaics and see what those signals look like. So there's like a whole area in this that needs to get explored, I think. Because those two maps, when I saw them, as crude as they are, and as they are using slightly different populations, they still had a crazy amount of overlap. And especially when I looked at the the northern South American, like, seemingly outlier is a seeming outlier on both maps. And that's really the intriguing part. Like,
that in itself is very interesting.
the overlaps with Denisovan DNA are strong in this, but so is the outlier, the dose-dependent sort of nature of Neanderthal contribution to the global population.
How much of the genetic percentage is made up?
And affecting current modern human penile size. Yeah. As I said it, you're right.
You did it?
Zombie
Yeah.
Tom Hanks. Global what size? Yeah.
Yeah, you heard him.
That's what we're talking about.
But I mean, we come back to this a lot. Morphology is influenced by genetics, by environment. There's a lot of these things. And as we get into questions of why humans are distributed, adapted in particular ways, Like, how do these things all interplay?
Yeah.
How does it all work together? It's an interesting observation you've got going on there. I hope that you are looking through the scientific literature to see how this has been addressed in the genetic.
So the one thing that sort of led me down this new Neanderthal is a hybrid with Homo erectus, because that sort of mule scenario that I started us out with. If Denisovans are the mules, it really doesn't answer a whole lot of questions. If Neanderthals are the mule, and we've taken their genome and called it Neanderthal, and now are seeing anything else that looks like that one actually it answers a whole lot more questions, and it fits the model of it. You know what this reminds me of? This isn't like this portion of it, the initial like I should look into this, is sort of like when a kid looks at a map and discovers plate tectonics without being told, because you can see this is large puzzle pieces. They fit together. That's it. So on the surface there was that, and then I go back into research, and there's like, oh, Denisovans and Neanderthals split about 700,000 years ago. And also we found a yet unidentified archaic signature at the same time. Wait, what? Well, how can it be unknown? There's only so many things that we knew to be on the planet that there's an archaic unknown.
Well, it's like it's a subspecies, a hybrid, another species. Yeah, it's the braided stream. Everything came from origin and split and rebraided and split and rebraided.
Branch and a combined branch.
It's math.
When you look back, when you're doing the molecular count back to when these things were the same, are going to look the same. Is my point. Plus the fact that they have an archaic that kind of, then it's like a split with an archaic, which makes sense. And my initial thought was that means what we're calling Denisovan is the branch that changes from Neanderthal because of this archaic influence. But now, the more I've thought about it, I've come to the conclusion that the split is Neanderthals from the human shared ancestor, which would have been in Africa and out of Africa at the same time, and would have stayed itself for hundreds of thousands of years before humans come along, which it either develops into us or we split off from it at some point later. But it's one of those weird things, like when they talk about like when humans and Neanderthals diverged, we didn't take a separate path and then there was nothing. They branched off, and then that thing that they branched off from stayed itself in Africa for a long time before we show up.
Yeah.
Okay. So it's braided stream, but it's also sometimes the thing that you split from continues. So then it's just 3 branches instead of 2. But the whole thing then makes way more sense if that archaic interaction is what leads to Neanderthals, and that the archaic itself remains itself and is now what we're calling Denisovan. And even, there's still archaic that we keep seeing signals of current modern human too that hasn't been associated with Denisovan or Neanderthal yet. But I still think it is all answered by Denisovan DNA being Homo erectus and is the archaic. It's only that we've got it separated because we don't have overlap with what we have seen in Neanderthal, which we defined as a whole. It's a thing that's Neanderthal, and they have a separate evolutionary path and never combined. And the idea that they never combined for hundreds of, out of leaving Africa, even though they combined immediately when current modern humans show back up, just kind of wouldn't make sense. And 700,000 years ago, by the way, too, is, they wouldn't have been as removed from Homo erectus as evolutionarily as we are now, because this was like a long way back in the almost halfway back into the history of it. So
this is fascinating. I found an article from 2000. I don't know. When was this published?
you gotta be careful because you go back too far.
Oh, 2021. This article is from 2021. Apparently, looking at DNA from an individual who had like a great grandfather who was Neanderthal. They had looked at a number of things in the DNA. So apparently, and I did not realize this, our closest living relatives, the common and bonobo chimpanzees, have penile spines, barbs that chafe the vagina. And apparently, not only is human DNA lacking the genes for those spines, but also our Neanderthal and Denisovan genomes.
Yeah.
So this is why there's speculation that there's monogamy involved, because the spininess is good for promiscuous species. Didn't know that. Yeah. This particular evidence as well, there's androgen evidence suggesting that Neanderthals were more promiscuous than humans.
Okay, that would surprise me.
I don't know,
surprise me.
Yeah,
With
they're basing it off of that, the digit ratio, like the length of your fingers and the way that androgen, testosterone impacts your finger growth.
Yeah.
So apparently, because of that, their Neanderthal ratios suggest that they were more promiscuous because they had more testosterone than humans.
More testosterone. Okay.
Yes.
Well, they might have needed it.
Fascinating article here.
But that doesn't. But yeah. But then But does it? Does that say anything about? That wouldn't say anything about length of time of that growth, though. See, this is like if they reach adulthood by age 14, compared to humans, it's something very accelerated like this. It means they missed all those hormonal teenage years where the bulk and girth of growth is taking place.
I don't know.
Yeah.
Yeah, it's fascinating. Researchers have been studying HPV and stuff related to hepatitis related to Neanderthal DNA and strains that the DNA react to, antigens and things. Additionally, they've been looking at oral microbes left because of the teeth and what's left, the DNA of microbes left in the teeth and mouths to suggest that maybe Neanderthals and humans kissed. Because there was a
they're humans.
Kissing isn't necessary.
Wow. Okay.
Gross.
Good to know. I just think kissing's cool. Like non-kissing cousins. That's what would be Neanderthals and humans otherwise. Yeah. So we're kissing cousins.
There's some interesting stuff in this history. This history that researchers are digging into various different things. Neanderthals, Denisovans.
There was one of these Neanderthal finds, and I don't think it's the one. I don't think it's the finger, but one where it was. They were like, "This we have found a Denisovan that, or a mix, a hybrid that had two parents, one that was Denisovan and one that was Neanderthal."
Yes.
What are the chances that you found an individual happened to be the direct descendant out of 3 or 4 Denisovans that we've ever found and gotten DNA from, that you found one whose parents were both Denisovan and Neanderthal, compared to what you've been calling Denisovan and Neanderthal. One is a hybrid already, and so when you
It's the same possibility as finding a kid's toy.
Yeah, no, I think it's a lot. I think that one alone almost proved my hypothesis that Denisovan is 100% Homo erectus, and the fact that they're calling half of its genome Neanderthal would make it so that they would find a bunch of these eventually that had both parents, one a Neanderthal and one a Denisovan. In fact, it's probably going to be all of them that they find that look like they're the first generation of the split every time, as though Neanderthals and Denisovans solely sought each other out in the ancient world to mate and never mated with their own kind in Asia. Ridiculous, I say.
And that is just the way it is. The just so stories. No, yeah.
I need to
dig into all the hypotheses.
I need to get a server so I can play with the bioinformatics of the genome.
Yeah. You don't need a server. You just need access to a cloud, to one of those services that do it.
I've got the genome ready to go. It's huge. The Denisovan Neanderthal genome is huge. I don't think I could put them on my computer and do anything with them. I think I want.
You need a bioinformatics specialist to look into it.
I have one. I just don't have the server.
Processing power.
We need to bring somebody on who can, by the way, shut this down before it goes further, because it's going to consume a part of my brain that won't let go of it.
You've already it's gone there. You've grabbed on. You want to know.
What did I say? Current humans and Homo erectus directly mated in Asia? Is that what I'm saying now? I think that's what I'm saying now. It sounds, people laugh all you want, people. But I was right about the Neanderthals and the current modern humans, and I'll be right again.
Or you'll be wrong, but it's always good to
I could totally
make a guess, right?
Make a guess, make an educated guess. Try to make it an educated guess. Try to dissuade you. But I did. I went and I kept looking for. Okay, so the way that they've determined the genome obviously is going to tell me that they've already looked at this, or they've already figured out that this isn't correct. And it turns out that when they're backtracking, the Denisovan and the Neanderthal genome, they're finding that they split 700,000 years ago based on the fact of how many additional changes have happened to those genomes over time. However, how would you disassociate that from a merging or a mixing? And
they're doing it. It has to do with proportions. It's all the math, right? Like right now, it's like mutations and differences, and it's math. It's
a 200,000 year window, which is kind of big. So it's not precise, but it's
not precise, but there are known rates of mutational change. There are.
Oh yeah.
But there's like it's
absolutely.
just. Yeah, but I'm just saying that. The reason what they've come up with right now, their hypotheses and everything, it's not just it's math is what they're using. They're looking at the number of differences and the ways things that it's proportions and frequencies of things showing up. So
and what it does is it traces back to 700,000 years ish. Right. Yeah. But once you're there's not really an explanation of how that starts. If that's a splitting or if that's a merging. There's no way of disentangling that, which I thought somehow that there might be, but I haven't seen it at least. There may be.
More math.
Still, more explanation.
More math.
But
But that's why they have these ideas of like unknown human species that we haven't found yet. Like it's because they're trying to come up with different hypotheses to explain the relationships and why the math works for a certain branching of the phylogenetic tree. Like why do certain things appear to be more or things, human ancestors, appear to be more or less related, right? How is the braided stream braiding? And so that it's
But again,
it's all a math puzzle.
if you're trying to identify a horse and all you have listed categorized is a mule, then that horse is going to look like half a mule. If that's your reference, that's the problem.
Yeah. I need to go to bed now so I can get up in the morning and take my son to school and do the parenting thing, you know. Say good morning, Justin.
Good morning, Justin.
You are bright and shiny and ready to go.
That's because I drink breast milk.
I know you're... Pause from a...
And I'm ready to go.
And what do you say to me now?
Good night, Kiki.
Good night, everyone. Oh gosh, I'm breaking my lamps. I hope you have a wonderful week. We'll be back here again next Wednesday. Thank you for joining us for this episode of This Week in Science. Stay safe, stay healthy as you can. Get your sleep if you can. Stay curious, and of course, stay lucky.